I'm always interested in in the unknowns, and looking at the Great Lakes, there's a lot of assumptions that we know everything that's going on out there, which really isn't the case.
There are a lot of really important linkages between what's happening in an ecosystem ecologically, such as the Great Lakes, and the impact that that system has on communities such as Milwaukee that rely on the Great Lakes for economic growth and food and recreation.
It's just been a really interesting experience, just discovering really a new realm of learning for me. My background is just in ecology as a whole, and more terrestrial things on land, not so much in the water, but I just kind of discovered a few interesting classes here at the School of Freshwater Sciences, and they have just really piqued my interest.
Not to mention that the Great Lakes keep on changing with invasive species, et cetera.
So when we're talking about invasive species can change the way a whole system works. It can change what's being eaten, what's in the lake for food, what the water looks like, how nice it is to swim in or boat in, and understanding all of those things can improve management practices, which can directly impact communities such as Milwaukee.
We don't know a lot about that intersection between lakes and rivers, especially when you look at the Milwaukee estuary. There wasn't a lot of information on what they found were habitat hotspots. It turned out that these areas were relatively disjointed, right? So not a lot of connection between one hotspot to the other. And the hope is to improve these habitats and be able to provide corridors or improve the habitat between the hotspots, so that we can get better habitat overall.
I'm working with the Harbor District and the School of Freshwater Sciences to build these habitat hotels, which are old fry baskets actually for fish fries that we've repurposed to plant plants in to help little baby fish have a habitat to survive. Along these steel walls, there's not really much habitat for them at all. It's just pretty much a lot of mussels.
I work with two invasive species, primarily quagga mussels, which are a small mussel that attaches to rocks and substrate on the bottom of a lake. And then also round gobies, which are a small fish that also live on the bottom of the lake and eat the quagga mussels. So my work primarily is looking at kind of the interaction between those two and how round gobies, how them eating quagga mussels is impacting other fish that we enjoy, such as lake trout or lake whitefish.
I'm working on mapping the habitat for 4 harbors up the Lake Michigan coast on the Wisconsin shoreline. It's Port Washington, Sheboygan, Two Rivers, and Manitowoc. Looking at what habitat exists in these harbors, because it seems like something that should be known, but it's really, nobody really knows what's down there.
We primarily work at 2 different sites, one near Atwater Beach in Milwaukee, and then across the lake in Leland, Michigan, off the shore of Sleepmere Dunes National Lakeshore.
We go out with a boat and we drive transects around the harbors. And the entire time we're conducting the mapping, we're using a sonar to get basically the depths. You can get an idea of what the bathymetry is of the bottom, and that's essentially the topography underwater.
So once we bring in samples, a lot of our measurements are just looking at how many we have, the size of them, how much they weigh. And then we also look at things such as how old the fish are, and we analyze scales or small ear bones called otoliths in the fish to determine how old they are.
So what we're doing right now to monitor them is we're taking this camera on this pole and kind of just sticking it down and taking some video every week to track how the plants are doing and what kind of fish are hanging out around there.
Actually using a side scan imaging sonar. So we're looking left and right, and we're essentially taking an image along that path.
We'll also look at what's in their gut, or in their round gobies don't have stomachs, so what's in their gut content, and also the stable isotopes, so carbon or nitrogen isotopes that are in them, and that can tell us a lot about what they're eating. And connecting all those pieces tells us how a species is impacting an ecosystem.
Stitching those mosaics together gives us a general picture of what each harbor looks like. And then we go back and after interpreting those images, we say, well, we're not sure what this is. This looks very interesting. Let's go check it out, whether it be with an ROV or through scuba diving.
I love being this close to the water. So the University of Wisconsin-Milwaukee has the unique advantage of being located literally feet from the harbor. So it's really easy to go out and collect samples.
And yeah, it's just really important to have habitat to help repopulate the perch and all the other fish population in the lake.
We could say, hey, look what's in your harbor. Isn't that great, right?
Doing field work was definitely a draw for me. And Harvey's lab does a lot of work in the field, including a lot of diving. And I was dive certified before I came to the school, which I think helped and gave me an opportunity to work here.
My fish class just really got me just really interested in all different fish habitats and underwater like plants and stuff. And yeah, it's just been really awesome.
John's brilliant, right? And he's probably one of the top fishery scientists in the Great Lakes.
Harvey's very involved in a lot of different projects, and he knows so much about the Great Lakes and freshwater systems in general, and has just been like a fantastic resource and mentor throughout all my time here.
It's amazing to be able to work with somebody that has such a wide breadth of background and inherent knowledge that comes with that. He's playing 3-dimensional chess when you and I are playing 2.
So talking to him and knowing that I would have someone to answer questions and talk me through things and really help me along in the process was the primary reason I chose UWM.
Been a great person to learn from.